Have you ever wished you were taller, or you weren't colorblind, or you had a different color of hair? Well, the new biotechnology CRISPR makes all of this possible. It allows you to go in and alter the human genome. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a new technology which utilizes enzymes created by bacteria to split apart DNA. It splits apart DNA between set pairs of nucleotides depending on which enzyme is used. It can create sticky ends or blunt ends depending on what's needed by the researcher and which enzyme they decide to use. Sticky ends look like this, so they're unevenly cut on purpose to orient the correct way because they won't bind like this. However, blunt ends are cut right between two sets right across from each other so that they fit right together like this. They're a lot more universal, but they can also get orientated the wrong way a lot easier. Cas9 is the enzyme used in most CRISPR processes. So the really cool part about this new technology is from this split DNA, genes can either be added or entirely removed. The genes that are deleted in our current technology are ones that are malfunctioning in some way that cause harm to the individual, such as HIV and sickle cell anemia. Another option, if the gene is involved in too many functions or it cannot be removed without damaging DNA, is to add a properly functioning gene to counteract the non-functioning one. So the second gene will work properly, and even though the first one isn't, it will create the necessary genes to keep the individual healthy. So because proteins are created based off of the genes, which are the blueprint for the body, and the proteins are the building blocks for the body, by changing the DNA, you can dramatically alter the way the body functions. Because the proteins, if they function correctly, means the whole body functions correctly. Here is a demonstrative analogy. This is our DNA. It's the set of instructions used to build proteins. This is our mRNA. mRNA can only follow what is directly written in here, and he can't make any choices for himself. In this case, a step was missing, so mRNA built a not quite functioning protein. The roof isn't aligned, so it can't keep any water out. With using CRISPR, we can chop up the DNA. This is a CAS enzyme. And we can use covalent phosphodiester to insert our new gene. We can add this new gene. And now, next time mRNA builds it, he has the proper steps to build a properly functioning protein like this.